Well, good evening, everyone. It's so lovely to see people in a lecture theatre. I haven't given a lecture in person for a long time. I don't know if Riley has, but clearly this is a great group to be giving your first lecture in a few years to. My name is Anthony Bull. I'm head of the Department of Bioengineering, and it is my pleasure to welcome you to the inaugural lecture of Professor Riley Green. Don't clap yet. I'm going to say just a few words before we start. I'd like to specifically welcome Riley's family in person, so many of you, wonderful. And if you hear any noise from the corner here, they're just living up to the family's name, okay, and they're there, and they're going to be participating in this lecture, I think. There are also many family members remote online. I gather it's 2:30 in the morning. Thank you for getting up. But I also gather that you can watch this later on because it'll be recorded. And of course, welcome to previous PhD students. First PhD graduate from Imperial College has come back from Germany. The research group, which is covering quite a few seats at the back, and many others, collaborators, former group members. And inaugural lectures are this kind of family occasion. It's where we, the family, and the family is broader than just this family, the bioengineering family, the family of this institution, the academic family, celebrate and acknowledge one of our own, who's achieved the title of professor and is recognized as an expert in their field of endeavor. Riley joined Imperial in 2016. That's not very long ago, you may say, as a senior lecturer from the University of New South Wales, and where she had been a postdoc, a lecturer, and a senior lecturer prior to joining us. But she also did her mechatronic engineering bachelor's, her biomedical engineering master's, and her biomedical engineering PhD there as well. So she was there for a long time before she came here. And then in 2018, yes, that's very soon, promoted to reader, and then in 2021 to professor. She doesn't hang around. Today, Riley will present to you some of her outstanding research and some other things. But I want to highlight that Riley is a colleague who breathes with both lungs. And what I mean by that is she breathes with the lung of research and translation and the lung of teaching and broader contribution. And as any expert on lungs will tell you, and I'm not, she's more of an expert on that, that you can survive on one lung. But actually, by exercising, you do so much better with both lungs. And they actually interact as well. And maybe that'll come out today. So I'll just highlight a few of Riley's other contributions, the second lung. And for many of us, that's the first lung. She's a highly valued member of the department. Her teaching is excellent and broad. So I'm just going to read out some of the courses that Riley has taught here and in University of New South Wales. Molecules, cells, and processes, statistics and computational methods, brain-machine interfaces, mechanics of biomaterials, cardiorespiratory system, she's clearly more of an expert than me, and there are more. So this is someone who has broad expertise and is actually very willing to teach in these other areas. She's also taken on a number of key roles in the department that have produced fruit in so many different ways. For example, Riley, very soon after she came, she ran a project for us as academic lead to figure out our safety systems as we were transitioning from one to another, and so has contributed very much to making things better, easier, and safer for the whole department. She's proactive. As a small example, I remember when we had a very significant strategic equipment bid, and we had to have 3 people go and be interviewed at the EPSRC for this. And there was the PI, there was the institutional representative who had to come along, and I was going to be that person. And then the third person had to be somebody who was excellent, who was a potential user, who could represent the broad use, and who would be very good at interview. Riley was the right person, but she was on maternity leave. Without a single hesitation, Riley volunteered because she thought she was the best for it, and clearly she was brilliant. We won the bid, and it's just so clear that she's so collegiate and supportive of everybody here. And her excellence is recognized in other ways, but also, you know, she was an elected member by her fellow academic staff in the department, demonstrating her standing within the department. She's good at coming alongside others. She's a mentor and a supporter. She leads a thriving research group, and all of those individuals will testify to that, where these individuals go on to do wonderful things and are doing wonderful things even while they're here. And that capacity has been recognized in the department, where she has been a line manager of academic staff for the past 3 years as well. She's now embarking on a very significant new phase in her career, and ours, where she will take on the head of department role from the 1st of August. And we all look forward to that. But for now, we've been looking forward to your inaugural lecture. And so, Riley, I'll hand over to you.
Thank you. Okay, thank you all very much for joining us here today, and looking forward to taking you through a journey of my research career and my time both in Australia and here in London. First of all, I just wanted to talk a little bit about cyborgs. So we're really just talking about a blend here, the blend of man and machine, the bionic man, anything that's organic that also contains biomechatronics. And so we cover a really wide variety of different devices, and I'm going to walk you through my journey as we walk around our bionic man. Firstly, my small declaration of interest is that we are now trying to translate some of this work through a company in an effort to make sure we're making more impact as we develop these exciting technologies. So now we're going to start way, way back in the 1700s, when there was a couple of experimentalists who were trying to figure out electricity, having quite a few arguments in the literature. And one of them was Luigi Galvani. And he used to like to go out in thunderstorms and hang frogs' legs off fences in an effort to prove that body tissues had their own form of electricity. Not to be outdone, Alessandro Volta, who believed that only metals could contain electricity, and he built batteries that he subsequently applied to his head, creating what might be the first brain-machine interface. It didn't go so well. He described it as a jolt to the head and a sound of, I think, bubbling dough, which I think might have been his brain. Interestingly enough, both of them had their merits, and today we understand a lot more about that. Run forward about 200 years, and we're now at a point here in the 1950s where you can see one of Medtronic's first devices, which is one of the first mobile cardiac pacemakers, which has returned amazing quality of life to people across the world. So we've come a long way. What can we do now? And this is just an example of, again, Medtronic and where they've come in merely the last couple of decades.
With your index finger to your nose, back to me, and to your nose,
just do the best
and back to me. Good. Now, with both hands at the same time, can you tap your thumb and your index finger as wide open and fast as you can? Pretty tough. Can you, with both hands, can you open and close?
Look like entirely different people.
Able to do that?
Good.
We can do so much now with electricity, with the devices that we've developed. But when we talk about bionic organs, when we talk about bionic ears and bionic eyes, we think that we can reflect and replace the bits of the nervous system, the nerves and the brain that we have lost. But we still have major challenges. So I'm going to play you what is a cochlear implant simulation, and I want you to have a good hard listen to this and tell me if you can pick which music track this is. If you've already heard it, and I know several in my group has, no cheating. Not great for dancing to. Does anyone have any idea what music that might be? Not you. Let's try again.
It's a beautiful day.
Still have a really wide divide between what the normal nervous system can provide and what we can replace with simple electrical devices. And this is where my journey starts. So I started off, as Anthony mentioned, doing my degrees and my start of my career at the University of New South Wales in Sydney. absolutely beautiful location. This here is a picture of the Graduate School of Biomedical Engineering, where I did most of my work. And it's exactly 7 minutes from the library down to the beach. I knew that very well during my undergraduate. And as time went on, less and less time was spent on that shore, but it's still there. During my undergraduate is actually where I first started working in bionics. And Bionic Eye is the project that was, for anyone who's at their early stages of their career, it was my undergraduate project. I was working on a small component of that, which started in 1997. And that's when this guy here, Greg, who was a Cochlear employee, came to the guy who was eventually my supervisor here, Nigel Lovell, and you'll hear from him a little bit later. They started this group with the idea that they could do for bionic eyes what had already been produced for bionic ears and cochlear implants within Australia. So the aim there was to restore a perception of sight, and we say a perception of sight because we know that's really difficult to restore full nervous systems. And this was for patients who had a disease that had caused the light detecting cells in their eyes to die. So small patient base, but a really great way of proving up a technology. So they worked within the university with this shady crew from 1997 until about 2009. And then we were successful in 2010, at which point I was a postdoc within the group of receiving $50 million of funding from the Australian Research Council and a much, much larger group that ended up working on that by 2015 to take these bionic eye technologies, get them into the clinic and start to do patient testing. And it was an amazing experience. Along the way, we met many patients and started to feel out how you're actually going to get one of these devices and what people could get as benefit from them. So a little bit about the actual technology, because a lot of our bionic systems are built off this same platform. So here we have a camera that picks up an image, basically your normal video, that's then processed down on a body-worn processor to a number of spots of light, effectively, and each one of those spots of light is an ability to perceive part of the image, but it's only ever part of the image, and you've got to kind of bring them together to make a full image in the brain. So this is where my area started to focus, which was this little area right in here. It's an internal implant of effectively, they're called electrodes. It's how we stimulate the cells using a contact point to the nerves that are left in the eye. So each one of those has the potential to produce a spot of light. And the question we had throughout most of my work was, how many electrodes do you actually need to produce a useful image? We started out with the idea that we could produce one with 98 electrodes. And this is a very difficult image if you don't know what it is to try and realize and figure out what it could be that you're trying to look at. To put this in perspective, the current leading bionic eye on the market is a company called Second Sight at the moment. They're in hundreds of patients worldwide, and they have 60 electrodes, of which with most patients only about 40 work. So we're still quite far away from being able to produce really useful images. And part of when we were working on this, we spoke to patients who were blind, who said, if I am going to go into a trial, if I'm going to accept one of these devices, I want to be able to do something more than just know where the big objects are around my house. I can already do that. I want to be able to see my children smile. I want to be able to improve my social interaction. So I want a lot more out of the device you're trying to produce. So you could do 200, and you're getting a little bit closer. 600, and we might start, if we squint to see what's going on. If we can get up to about 1,600 electrodes, we start to see that this is actually a person's face. And that's kind of the level of, of resolution that they're going to want. So this was the challenge, is how do we actually get this many spots of light, this many electrodes into somebody's eye, which is a very, very small space. Roughly about 3 millimeters is the central vision part, and it's very hard to pack electrodes in there. Not just physically. Now we're going to talk about how we've done this in the past, and why we're still doing it with the same materials to this day. Cochlear implants were made in the 1970s with 22 platinum electrodes. This day, the top electrode system is still a cochlear implant with 22 electrodes. So we can do more, and we need to push these technologies. They exist, and they've been doing so well because they have an amazing history of use of being known to be safe, and that they do enable amazing ability for people to do things like learn to speak in cochlear implants. Even in Bionic Eye, people are finding they can read really big letters. So we have a lot of use for them, but we can do better. And the reason why we have a major problem is because metals talk in electrons, Volta style, and cells talk in ions, Galvani style. So we need to bring them together. Whenever we actually stimulate tissue, we go from the metal with electrons and have to convert it to ions that those cells can do something with. So that's a challenge, because what happens is the electrons get to the end of their path, they see the body tissue, and then they have to change something in it to make it into an ion. When they talk about something like vision, what you can see here in this graph is all spots over here which correlate to the amount of electricity needed for one of these patients who was actually blind to see a spot of light. So you can see there's a wide number of patients with all different levels that they need, but a huge number need quite a bit of a charge to actually see a spot of light. Down the bottom here is a little bit of data we collected that shows the safety limit and how much electricity you can put through a metal electrode before you start to get changes that will damage the tissue. So they will change pH, they will generate gases within the body and destroy the electrodes themselves. They also can heat up over time if they're driven too hard. And so they have some major drawbacks as well. The next thing is that metals are very, very hard. They're inorganic, they're quite stiff, and our brain, our nervous tissue is very soft. In particular, this is from our friends at the Science Museum, our brain and all of our nerves jiggle around like this all the time. With each heartbeat, they jiggle. And so this soft, jiggly material moves against this hard, stiff, inorganic material and effectively irritates all the cells in the environment and produces what's a scar. Exactly the same as this, but inside in your brain, on your nerves, and they stop working the way that they're meant to. So we start to create damage in a system that's already diseased and injured. So these are the things that we look to fix, and this is where I spent most of my PhD time, working in a material called a conductive polymer. So these little guys are great, because these guys carry electricity both in electrons and in ions. They can move seamlessly between talking to hardware electronics and talking to tissue. And they do a really great job at mediating that. They're organic, so the body doesn't immediately try and kick them out. And they have these nice little rough surfaces that cells can grow into and interact with. So they have some major benefits. We also have this extra thing here that says they can be biofunctionalized, which means we can put proteins in them, things that are part of your body, to try and trick the body into thinking that they're part of it and not reject them. They're softer. They're not as soft as nerve tissues, but they're softer, and that sort of helps mediate that difference as well. However, it's a PhD, so it can never go easily. And what we demonstrated throughout most of the PhD was that this is a common problem. As you start to mediate that difference in stiffness between soft and hard, you create another layer that's prone to falling apart, that creates that disruption between the polymer and the metals. And the more you try and encourage biological components in there, the more things you put into that material, the more damage you do to the actual material itself. You lose the electrical functionality, you lose its ability to be robust and stay where you put it inside the body. So, during my PhD, which had a very complicated title, could be very easily interpreted as how to make conductive polymers less conductive and fall apart. It's a common experience, but out of the ashes, and for all the PhDs in there, there is always an out of the ashes. We developed something called conductive hydrogels. We took all the things that we'd learnt from all the failure and all the problems and found a way around them. So we developed these things called conductive hydrogels, which took our good conducting polymer friend here. PDOT is one that's very common in the literature, that brings our electrical performance, and combined it with something that was more robust, that was softer. And this is a hydrogel. The particular hydrogel we used was something called polyvinyl alcohol, or for most of you, it's the material we make lots of soft contact lenses out of. So soft, squishy, movable, and we combined the two to make a hybrid material that we subsequently patented, and have used and looked at with many of our devices. So revisiting our Bionic Eye, if we then look at the safety and how much electricity we can put through our devices when we coat them with our conductive hydrogel, so just here, one of the same ones we were looking at before sitting right beside it, we were able to finally show that our safety limit here with these materials applied to the exact same device was much higher. So much so that we can now actually deliver safe stimulation and vision to patients. We could easily make our electrodes into the hundreds and deliver a higher resolution of vision. However, in the short project time that we had, we ended up going into patients with just platinum electrodes, but it was still an amazing learning experience. So I'm just going to tie this one up with our much smaller electrode array than we were possibly capable of. And the reason why is because whenever you develop a high resolution system, you actually need the connections to that. You need the electronics to drive it. You can't just develop one part. So that needed to be brought back and develop the other parts of the system before we could move it forwards. But we still wanted to learn something from patients. We still wanted to figure out during the course of the project what benefits we could have to our blind population. So just a brief video.
Diane's doing experiments where we're having a set of obstacles in front of her on the ground, and she needs to move from the back part of this environment through to the front part, and we're looking at how long it takes her to pass through that pass and how many collisions she has with obstacles or brushes against the wall. So she's wearing a backpack computer, which is attached to a head-mounted camera incorporating a depth sensor, and the backpack is processing that and then doing the vision processing of that and then putting up the set of things that we want to have stimulated on the electrode array for her to then gain an impression of what she's seeing in the image.
Oh, this is, you can see a reason for the Bionic Eye. You can see that there's a practical use for it. It's just, it's really good.
Properly Australian, really good. What was really great about actually having Diane as a patient was Diane is one of these patients who has the right choice for a clinical trial, a pilot trial. She has a daughter and a granddaughter, both of who suffer from the same condition, who are not yet blind, and is a great choice for somebody who can give you lots of feedback. She was doing a PhD herself, and has been a great source that still interacts with bionic vision to this day. Through our work on bionic vision, we obviously came in contact a lot with, through the Bionics Institute, which is in Melbourne, who was part of the Bionic Vision Initiative, and where the original cochlear implant was developed by Graham Clark. We worked a lot and came in contact with Cochlear. So we then started to look at how our materials could be used to help improve not just vision, but also hearing. There we go. So this is a cochlear implant, just up here. And this is what it looks like when we coat it with our conductive hydrogel. Most of our materials, they kind of all look the same when you put them in a picture. They do many other things on a microscopic level, but nice little dark bands here mean that we've got this jelly-like, contact lens-like coating all over our cochlear implant, conductive just on these little sensor sites here. The reason why I wanted to show this data was because when we work with polymers, which they're effectively plastics, right, people start to ask you, well, they can't possibly be as stable because they're plastic. They couldn't be as stable as metal, and the answer is actually, when you're driving them electrically, these guys are more stable than metals. And what you see over here is a study we did in simulated conditions with the fluid from inside the ear. And this is the power that's being required to drive the electrodes, or the impedance, how it's resisting the passage of electricity as you push it into in and out with stimulations typical of a cochlear implant. And this down here is the exact same data for our conductive hydrogel-coated electrodes. And what you can see is they're consuming less power, and they are very, very stable. They stay stable across the whole 2 billion stimulations, which is roughly 2 years of cochlear implant use. So we know that these materials are quite stable. We know that they have lots of capacity for improving the properties of these devices. We worked with Bionic Institute for a lot longer, and this sort of bridges some of the work. This was ongoing in the lab after I left. Down in the Bionics Institute, they were able to show that this was maintained even in an implant environment within actual cochlears. These are our safety limits, and they're saying the safety limits of our cochlear implants is much higher at the end of 5 weeks, even with some scar tissue growth. than it is on our platinum electrodes. So we know these have some great benefits. They really help us push electricity through into the tissue in a safe way, and we can apply them to lots of different sorts of devices. Also, the hydrogel material, our contact lens material, is much softer and has less of a reaction with our tissue. Unfortunately, these also still suffer from some of the drawbacks you see with coatings. Whenever you coat a device, there is a potential for some sort of failure, and this here is some conductive hydrogel that's gone off into the tissue and shows us that you still have a problem there where if you can't get a robust interface, if you have a lot of movement, you can lose material. So it's a great technology as a coating technology, but we kind of wanted more, and we wanted to expand this research and try new things. So we hark back to my story, and this is where we moved from Kensington in Sydney, where the University of New South Wales was, to Kensington in London, where Imperial College was. And all I can think of is the man who named the place in Sydney had never ever been to the place in London. Quite different. Both beautiful in their own ways. But throughout this journey and across the divide, we started working with a company that was starting up out of GlaxoSmithKline called GlaxoSmithKline Bioelectronics, and eventually became Galvani Bioelectronics when they spun out with Verily Life Sciences, who are the Google parent company. What they got us interested in was another area. This is peripheral nerve cuffs. So this is where you're actually taking an electrode and wrapping it around one of your nerves further down in your body. And they developed this area because they had identified that there are some conditions where you can treat them with normal drugs, pharmaceuticals, and you can keep trying and trying and trying, but you don't get a response from a patient. So they thought about a way that you could actually start to do that might be to block the nerve activity or stimulate the nerves in a different way so that you weren't getting that problem. Some examples are diabetes, rheumatoid arthritis, irritable bowel, and they looked at all these different conditions with lots of research partners across the world. They now have, or they're now well on their way to getting this and trialling it and getting it approved for rheumatoid arthritis, and they're having some great results there. But they got us really interested in how do we actually interface with these nerves, because they have different requirements. They wrap things around them, they try and steer the electricity into them to activate very specific points. And this here is some work we started at the University of New South Wales that then was picked up by a very talented master's student who I then would not leave and is still a PhD with me now, Estelle Coutaz, who basically were trying to do what we did with conductive hydrogels, but to make a device that could be all polymer, that could be stretchable, that didn't have any of those problems of coatings and metals. And we did that by developing these conductive elastomers that have our same conductive polymer friend here, but combined with an elastomer material. And we chose to work with polyurethane, which is well known in the medical implant world because it's used in a lot of other devices. So it's used in things like these, which are catheter lines, and also implantable devices. That's the outside of a breast implant that's made with polyurethane as well. So they're stretchable, they are easy to process, and they open up lots of different opportunities for making even 3D printed devices. So what we have here is, it's a sheet of this polymer material, which it doesn't make for an exciting picture, but the different one here is actually, this is a 3D printed version of some electrodes as well. So we have looked at different ways of processing them, and in particular, Estelle took them to the point where we could make full electrode arrays with them. So these are our little electrode arrays, and all the bits around it are all polymer, so that now we have a really stretchable device. If you did that with a metal device, it would just break. I took one of these to a conference and let everybody pull it apart all day long, and it still could conduct electricity at the end of the day. So we're really excited about these materials, and they formed a really great technology platform for us to start looking at lots of different things. In our own work, and with some funding from the EPSRC, we've been able to look at how they perform in sciatic nerve. And Dr. Umair Syed, who's also part of my group, sadly leaving us in a couple of weeks, spent a lot of time working out how to set up these experiments and how to show that these devices work really well. They work as well as our metal commercial materials, and even better when we look at how much charge we can safely push through them. So we know we're getting that really good performance in different tissues in different environments as well. Then we sort of started getting some inquiries, different combinations of collaborations, and as I started my work at Imperial, I was looking for different people that weren't the same things we've been doing in Australia. And we started a collaboration with the National Heart and Lung Institute with Professor Sian Harding. And this was looking at cardiac pacemakers. And there's a really nice alignment here between the sorts of things we do in materials and the sorts of things that they're looking for to solve problems with in pacing devices. Another one of my students, Alexei Novikov, took this on and did an amazing job of taking a really complex problem, trying to make long pacing wires that were all polymeric, that could stretch and remain conductive. They could still push electricity through them. He did a really interesting thing where he actually formed the PDOT, the conductive pathway first, and then embedded all this insulative material around it, but was able to, on the bench, I think it's actually really impressive, on the bench could show that these materials and these wires he makes can meet this regulatory standard for pacemaking that's set out internationally. So we know that we have conductivity that's maintained when we strain and stretch it, and when we make it the right way, not this way, we have something that retains conductivity over long periods of time. The great thing about this is that we have now a component which can work not just in pacing devices but in others. That means that these patients can undergo all the standard monitoring and diagnostics that any other patient might. So a standard cardiac pacemaker recipient has so much metal wires in them that as you put them in something like an MRI, generates heat and generates unsafe effects to the point where they have to be very careful when they are trying to do MRI to look at where the device is, to look at whether it's operating properly. If we were to use these materials, we wouldn't need that at all. You can throw them in the strongest MRI, get them the best imaging, the best diagnostics, and we're not going to have any problems from metals inducting heat or causing any other effects on the tissues. So this is a really exciting program. It's just in its infancy, but I think is a really nice new application that we're developing in. We've also been asked a lot about wearables. So we worked for a very long period of time, as you can see, in implantable devices, and that was kind of the standard. It's exceptionally difficult to move things in implantable devices through the clinic. You need lots of money, lots of driver, lots of support, and the regulatory bodies on board. Wearables presented a really nice opportunity for us to figure out whether we could use these things on the skin, outside the body. And so in particular, we've been interested in EEG, so brain signals, EMG, muscle signals, and ECG, cardiac signals. So they all look a little bit like this, and most of the time, what we do is we use these things called gel electrodes. They're wet and they require skin preparation. So every time these get applied in the clinic, someone comes along, they clean off your skin. Sometimes if there's a lot of hair, they'll shave it. Applying gels, or the gels sometimes come on the electrodes. They only work for a short period of time. They don't work forever. They need to be refreshed and replaced. And so we thought that we might be able to use our conductive elastomer materials, and we can use them dry. We've subsequently spent a lot of time with two of my postdocs, Bogachan and Karina, and Nick's a new PhD who's come on in this space, working together to make a few different iterations of these to find out whether they work on skin. So in particular, we have a collaboration with neonatal pediatricians, so newborn babies, to detect seizure. And the really important one for these babies is that if you suspect your newborn baby has seizure, unless they have a seizure between 9 to 5, Monday to Friday, they don't get diagnosed. They either get given anti-seizure medications that can cause lifelong effects, or they don't get given them and they wait and try and monitor it by sight. We're trying to develop for them these nice little fuzzy electrodes that we can place and anybody can place in the clinic that don't need skin preparation, they don't need a technical team, and can deliver hopefully life-saving technologies. We're also developing in collaboration with the US Defence Force, these little guys here, which is to monitor soldier vital signs and keep them sort of understanding what's going on the field and allow them to rapidly respond. This here is just a little bit of data that shows we can actually do this. These are comparing wet electrodes and our dry electrodes, and they have the exact same spectrum, the exact same signal coming through them.
And we can detect different conditions. So we can see when eyes are closed, we can see when eyes are open, they have different signals, and we're able to monitor those dry on the skin with no preparation. Although my caveat to that is we do often use the bald people in the group for this because it's a little easier barrier to get past. Thanks, Bogachan. So that's sort of the wearable space, and then we found this even more interesting space that we were invited by one of my collaborators up in Cambridge, George Maliaris, to collaborate with him on a drug delivery application. So this is where we're trying to use the same materials, the same devices, but we're trying to treat cancer. So the interesting part here is there are these brain tumours that grow that can't be removed. Some of them, they're either deep within the brain structures, or the way they grow with lots of little fibrils means that they penetrate all the different tissues, and so getting in there and doing any surgery is not any good. More than that, the only way they have of treating these patients is to dose them, their whole bodies, systemically with strong chemotherapeutic drugs, which poison the body in a hope of trying to poison the tumour. But the barrier that's around your brain that stops drugs from crossing it prevents large amounts of those drugs from doing anything useful to the tumour. And so they're in this really poor scenario where it is lucky for 5% of them to survive up to 14 months. So what we've been developing has been this little device that during a biopsy, we thread our device down there into the brain, and we use electricity to drive the drug out. It's dry, so it doesn't cause swelling in the brain, and the drugs can then be turned on and off and delivered in such a way that it's getting right into the tumour. And so this work's been spearheaded by Chris Chapman, who's another postdoc in the group. He's supported, whoops, oh, that wasn't right. There you go. He's supported by the Rosati's Trust on a fellowship, but we also have an incoming PhD student in the Cancer Research UK charity, and they're supported quite a bit with the EPSRC as well. What we've been able to do thus far with this is develop these devices, prototype them, and show that we have these model drugs, and when we drive it in one direction with electricity, we get release of one of the drugs, while simultaneously preventing the release of the other one. And then if we decide we want the other drug, we can switch that over, and we can drive release of that drug while stopping the other one from getting out, which gives us a really nice tunable system for delivering different sorts of drugs. My ultimate vision for this is to try and get it to the point where it's a chronic implant that a cancer patient can have in for 5 years, turned on and off at will to treat the tumour until they're sure that it's gone. So it's a long way down, it's a big path and a big project to take over, but if we can improve the life expectancy of these patients, which is dramatically short, I think, and some of them are children, there's a large number of children who get these brain tumours, that we can do some really good things. So we still have many other things going on in this space. We have some brain electrode arrays for doing things like implantable seizure detection. We have lots of high resolution options, and this is our good old friend, the Bionic Eye. We're still working a little bit in that space as well. But these are a great technology platform. Developing connections is something that Zach here has been doing, and we're finding some new ways of making these interact properly with electronics. But there's all the other development, as I mentioned, that needs to come along. So I'm going to pause this one for a moment. All of these devices and what we've been working with so far, it's all about electricity, right? It's all about pushing electricity into the body or detecting it and putting it into hardware. But when we do this, we're dealing with cells that are injured, cells that are diseased, so your body is in an unhappy state. And we think that electrocuting those cells to make them have a response is going to be a good thing. And then we get disappointed when they don't respond the way we think they should, that people aren't getting the best benefit out of them. So watching one of my favourite programs, and this is Firefly, which is a science fiction program from Joss Whedon, I saw this scene. And this scene is a spaceship that's coming on in the middle of space, docks onto another spaceship. And what you see growing out of here is the growing connection, right? It's coming out and growing in and plugging in itself. And I found myself wondering, why can't our devices grow into the brain? Why do we always wait for the body to respond to them? Let's make them living. Let's get our devices to go in and make connections that are natural connections, that look at that injured tissue and say, hang on, if I give you a nice healthy input and a nice activation that's natural, maybe you'll survive and hang around. Maybe we might even get some regeneration.
And that spawned an area mainly in brain-machine interfaces where it has its best application. But it's called living electrodes. And what we do here is we start with our devices, but we start to change them dramatically. We use tissue engineering or regenerative medicine approaches to put new materials on them, and we started off doing this with PVA, our old contact lens material, but we put stem cells inside. And then we drive those stem cells to differentiate into nerve cells themselves and try and make them create connections. There's lots of parts to this, and in our first effort, we did this back in Australia. We tried to do it with funding from DARPA that required we go from concept to proof of concept in 12 months. And it was a lot to try and do. It was quite a lot for these guys up here who, I think 6 months in, I left and came to Imperial, and they were holding things together back in Australia, and we had remote meetings, I think, regularly at 6 a.m. for me. But they did a fantastic job. And we got to the point here where we were able to show that we had a better signal that we could record in the brain with these devices. But whenever we looked at the cells inside these materials, they were not happy. These cells weren't producing nice neural networks. They weren't growing out and creating the nice connections that we had hoped for. Luckily, the ERC was nice to me and funded a project that's given us 5 years to do the same thing. And we're making some great steps. So at the moment, this project's being led by postdoc Roberto Portillo-Lara and PhD student Martina, who's been working on this now. She's in the final years of her PhD. What they brought to the table here was the idea that maybe the contact lens material, while quite nice, is still a synthetic material. It doesn't have a lot of goodness that, body things that would help it survive. And cells need to be able to manipulate their environment. So we need to give them the tools to do that. So we started working with this material here, which is a gelatin material. Most of you should know that as jelly, or if our American compatriots are online, jello. But what's great about gelatin is it has segments on it that cells can stick to, but it also has these little domains in it that cells can manipulate and modify, and they can create their environment to suit themselves. So when we started making these hydrogel materials for our devices, I was super happy when I saw this spread in one of my meetings with Martina. And what it shows is as we incorporate more of that gelatin, we get to a point where they go from unhappy to a little bit happy to, wow, our cells are finally creating networks here. They're growing, they're modifying their environment. And so to this day now, we have these neural networks being grown within these hydrogels, and we're putting them onto electrodes. These are all nerve cells, support cells, and they're growing into really nice functional tissues that we think we can now then progress into preclinical trials and show that this system can work. That's only one part of it. The other part of it is how do we tell the cells how to become the right sorts of cells? And so Sophia, who's been working on this project now for a couple of years, has been working on, if we're on a bionic device, why don't we use the bionic device in some useful way to stimulate and drive the development of these nerves into the right sorts of nerves, to make connections with the brain, to make connections with the environment they're in, not just within the device, but outside. So we want to grow them. And she's been looking at lots of different stimulation paradigms, and this has been done in collaboration with Nia Grossman up in the Brain Research Institute, or Dementia Research Institute in Brain Sciences. And what she's been doing, there's lots of complex data here, but the really important thing to look at here is this little column here, which is 10 hertz. This particular stimulation paradigm is really interesting. It's the exact signal that your brain makes when you're in utero to start neural network growth. So if we then do that to our stem cells that we have in our devices, we tell them that they're growing. We tell them that they need to make these connections. And so we found out that this is a really useful stimulation signal. And what we've been able to make from that, if you look at this now, this is what a live cell image, and each flash is showing an action potential. It's showing a nerve cell having a communication. And these guys are now talking. They're talking within each other. They know that they've become nerves. They have the support that they need. And they have the signals that tell them to become nerves. So we have really great building blocks here, and bringing those together and testing them is going to be the next steps. Really exciting project. We're really happy with the way it's going and looking forward to seeing where it goes next. And the last thing we're going to visit on you in bionic eyes, in bionic men or cyborgs, is a spawning project which we're just developing called a bionic spinal cord bridge. And this is where people have spinal cord injury. We'd like to take all the things we've learnt from living electrodes, all the things we've learnt about stem cells and turning them into nerves, and seeing if we can create an injectable system that we can use to grow new connections in a broken spinal cord. And so the idea here is, and Gillian Cole's been looking at this, is developing these self-assembly materials. That means we create one building block just here, and we then put them under the right conditions and inject them, and they form these nice little fibrils, which functionally look like this in the microscopy image. But the important thing, and the really interesting thing which Gillian's done, is added this nice little conductive element here. So this is our little conductive polymer, and when they align and when they assemble into these fibers, they should line up all those little conductive elements, so the cores of these fibers are now conductive polymers with a sheath of happy protein that then can interact with the body and help our stem cells grow. The reason why this is really a new way of doing it is because we think we should be able to address these using technology that our friends up in Brain Sciences in Nia's group have developed, which is temporal interference. It means I can stimulate these from outside the body, so I don't need an electronic implant inside the spine. I put the building blocks inside the spine and I stimulate it from outside and get our cells to grow and reconnect. We've got a lot of work to do in this. It's a concept more than anything at the moment, and we're just bringing together our building blocks, but we're pretty excited. So this is the end of it, and what's the future? My work, and I think the future here, is blurring these lines, blurring the lines between bionics, between tissue engineering, regenerative medicine, and bringing them together to make new devices, to address new problems that don't have good solutions yet. We want to make really high-resolution systems that can really reflect the nervous system, that can do what nature does itself and do it just as good, maybe better. To do this, we need to continue working with industry partnerships. We need to do that to get sort of supportive hardware that can look at the new way we're operating devices and create the systems that go with it. And obviously clinical partnerships to get our patients into trials and show that this is working the way we think it should. So that finishes it up for me, except the important part. The really important part, which is acknowledging all the guys who actually did most of this work. This team up here, which is my current team, a few of our alumni, there are many, but these are the few that work has been represented here today. Photos are much more fun than actual names. But first and foremost, Joe Godding has been with me since I supervised his PhD in Australia and continues to keep the show on the road when I've had 2 maternity leaves. And Olivia Calvi, who's a project manager who's been keeping me organised now for quite some time and keeping us all going in the same direction, which is fantastic. So thanks to those two. Next, the collaborators and our funders. So the local guys here, we have many collaborations, our clinical partners, the guys back in Australia, so Nigel Lovell and Laura Pulwarren, who not only supervised my PhD, but have been mentors. In fact, when I agreed to take on the head of department position, before I agreed, I spoke to Laura, who said, very sage advice. Ask yourself what you want to do. What did you think you were going to do next? And I was like, I don't know. She's like, well, ask yourself that and figure it out. Okay, let's see how we go. And then our industry partnerships, the work we've done with Galvani and our current work with the US Department of Defense, all great contributors. But none of that would be any good if we didn't have our support team. And so the bioengineering support team in particular has been amazing. The technical team, the teaching team, and the finance team who've helped as we integrate and get new people in, who make sure we all know what we're doing and what processes to follow. I couldn't find a swatch of the teaching team. I think that's missing. But in particular, I wanted to put a shout out to Ken, Keating, and Joel, who have definitely made things work for me in the lab and lots of my students. And also Pascal here, who had an amazing role when I came back from maternity leave with the oldest one there in tow. And every time he was shouting out in a research meeting, Pascal would appear and pick him up and take him away and bring him back to me asleep. He's amazing. If you ever need support, the electrical engineer in the tech team is the guy to go to. He was amazing. Lastly, but not leastly, family. My team, Emerson and Nara,
and my husband Ben, who has been a pillar of support for a couple of decades now, who allows all this to happen by working as hard as he can to support this endeavour going forward. My parents, who instilled in me the need for a good education, who continue to interact and help us in any ways that they can. And my parents-in-law who are here tonight, and a big shout out to them for coming from Australia all the way here for this talk. It is greatly appreciated, as is all your support over the years. And that's it from me. Thank you.
Wow. I'm not going to say too much wow. Others can do that. Wow. We're not going to ask questions now. We can do so later in person. I think that would be great. And so it's my pleasure to introduce the vote of thanks. A rather strange thing to do if you're not used to these inaugural lectures, but Molly will tell us all about that. She's Professor of Biomedical Materials and Regenerative Medicine in the departments of Bioengineering and Materials at Imperial College. She has awards and accolades that are longer than my arm in very small type font, so I'm not going to say much about that, other than she's a member of the US National Academy of Engineering, Fellow of the Royal Society, Fellow of the Royal Academy of Engineering, and many other things. More than that, she is a great friend and colleague, and so I'm going to hand over to Molly for the vote of thanks.
Thank you, Anthony. So I'm allowed to start with wow as well. So these inaugurals are always so nice. I was just saying to Anthony, actually, as we were coming in, an inaugural lecture is a bit like a wedding, isn't it? You get all your family and friends and all the people who you care about and who've supported you and who want to celebrate with you this really special occasion. So we're all really proud of you, Riley. I think it's wonderful and just such an exciting lecture as well. So you've heard that Riley just came here in 2016 and then became a professor just a few years after that, actually. So quite quickly, whilst also having children. I mean, really impressive. And one of the things I think that's a theme that might come up during this vote of thanks is that Riley is not at all scared of challenges. Riley's sort of fearless, isn't she? And, really kind of embraces everything that she gets into. And I think we're all very grateful for that science-wise, and of course also your kind of fearlessness in taking up this head of department role, which is going to be a completely different chapter in your life. So because Riley has obviously been trained in Australia, and if you haven't noticed is Australian, we decided that we would get some of the people who've been very, very special in Riley's scientific career to say a few words. So I'm going to try and plug this in and hope it works, because I'm not, believe it or not, known for my IT skills. So we'll see. I just need to unlock the password. Yeah, so there's going to be 2 short videos that I'm going to play. Now, the first one is from a wonderful person called Laura Poole Warren, who's a professor in Australia, and who I've discovered is ridiculously modest, because I asked her for a biography and she sent me a very simple biography. And then, of course, I looked her up and she's anything but simple. And in fact, Laura, as well as being sort of Associate Dean of Research, Pro Vice Chancellor, etc., has even been recognized as a member of the Order of Australia for service to biomedical engineering, which I think is about the top thing you can get in Australia. And basically, Laura has known Riley since her undergrad days. And when Riley came to see Laura about doing a PhD. Riley was actually the one that proposed her PhD project area to Laura, even though Laura wasn't even actually working in that area at the time. So again, not at all afraid of challenges. So I'm going to hand over to Laura now. I hope that's going to work. Is that coming up? So Laura, over to you.
Thank you, Molly. First, let me say what a great pleasure it is to be asked to give this vote of thanks for Professor Riley Green's inaugural professorial lecture. I'm so disappointed that I couldn't be there in person, but I can say with complete authority, despite having recorded this before I saw the lecture, that it was impressive. And I can say this because in the 20 or so years that I have known Professor Green, I have never seen her deliver a dud presentation. So, although my praise is somewhat anachronistic, I reiterate thank you, Professor Green, for your very impressive lecture, and congratulations on your promotion. To become a professor is a key milestone in academic life. You are now qualified to profess. And given that we all start this journey with a Doctor of Philosophy, I thought I might do a little bit of philosophical professing just to reflect on Riley Green's career up to becoming Professor Green. The work that Professor Green is doing is technologically brilliant, and it's in a critical area concerned with where electronics interface with the human body. So to move into the future of so-called bioelectronics, we're going to have to re-engineer them so that they're much more compatible with the body. And what Riley is doing is actually taking these materials and translating them into soft electronic constructs that are going to be able to conform with the body and to be much better integrated physiologically. This is a really ambitious area. It's incredibly important because it is going to help us design much higher resolution electronic devices, and it's going to allow them to interface with our bodies in a much more physiological way. It's a challenging area, and I probably don't need to add, for those of you who know Professor Green very well, that she has never been one to back down from a research challenge. If, or should I say when, Professor Green translates this research into neural interfacing devices that reach the clinic, I think it's a good time to think about what actually are the personal attributes that help you make such a success or reach such heights. Success is not just about the resources that you have. It's also about your leadership qualities. Over the many years that I have been lucky enough to work with Professor Green, and even when she was a PhD candidate in my lab, you could really see that she was not only demonstrating leadership qualities, but she was developing them and growing her leadership approach to problems. I think there are many leadership attributes that Professor Green has shown and really have underpinned her success to date. And I think I can sum these up in 5 words. And that's intelligence, innovation, persistence, resilience, and teamwork. And Professor Green, you have these in spades. You have achieved such great outcomes, and you expect from your team the same that you expect from yourself. And I think that these sorts of expectations include you take up challenges, and you really are keen on having very, very challenging research ambitions. You set the bar really high for success. And I think that comes with a corollary, which is you're not afraid of failure. And I think finally, you learn from your experiences. And these are things that you really embed in your team and has really underpinned where you are today. Again, I'd like to thank you for your wonderful lecture. I'd like to congratulate you on your promotion to Professor, and I would like to wish you all the very best for professing philosophically or otherwise into the future. Thank you.
Thank you. So there's a little more to come. So this was Laura, who I think put it so well about all the intellectual things that you've achieved, and also the sort of energy and bravery that goes with that. But there's another side to you as well, Riley, which is that you're, I think, a very fun and no-nonsense sort of person. And so we're now going to hear from another one of Riley's previous mentors, who's Nigel Lovell, who's also a professor. He was featured in one of the photos earlier. He was a previous president of IEEE, which is a wonderful organization and also very renowned. And he's going to talk also about your academic brilliance, but also about some other things as well, Riley. So let's see. Oh, no. There we go. So this is Nigel.
Very happy and honoured to add my congratulations to Riley Green in recognition of her professorial appointment at Imperial College. My name's Nigel Lovell, and I'm head of the Graduate School of Biomedical Engineering here at the University of New South Wales in Sydney. And like Laura, I've known Riley since she commenced her undergraduate studies in the late 1990s. Here at UNSW, followed by a master's degree in biomedical engineering and a PhD, and then postdoctoral positions in our school. Over the course of her studies and career, Riley has evolved into being one of the world leaders in implantable bioelectronics, soft biomaterials, and neural tissue interfaces. But I think we all know that Riley is much, much more than a world-leading scientist. Through a characteristic turn of phrase, she's vocal in her opinions, she's impassioned in her beliefs, and she knows how to build diverse and collaborative teams to achieve great scientific and technological outcomes. I certainly miss having Riley at UNSW as a colleague and as a collaborator, but I certainly don't miss what happens at nearly every conference where Riley and I happen to be in attendance. For some reason, we and our research group, with Riley as the tour leader, always seem to end up in some less than salubrious locations in the wee hours of the morning. Be it that gay bar in Antalya, where the locals tried to teach us Turkish dancing while showering us with bits of confetti made up of torn up table napkins, or perhaps in Osaka, where we started out with a meal comprising meat, sinew, cartilage, and probably every organ system of a number of chickens, before ending up at the Rock Bar, if I recall, in the wee hours of the morning again, at performing some air guitar impressions. So thanks, Riley, for some very notable and enduring memories. And those memories extend well beyond your undeniable research career highlights and recognitions. Let me wish you the very, very best from myself and indeed all the staff here at Biomedical Engineering at UNSW. I hope you'll continue to contribute greatly in the areas of flexible bioelectronics and electrode tissue interfaces, as you have done already. And I hope we can catch up soon, and given your new professorial title, maybe not in some sketchy bar, but let's try for a Michelin-hatted restaurant to the quiet backing of some orchestral music maybe. Congratulations, Riley. I feel very privileged to have been part of your very prestigious academic journey.
Thank you, Nigel. So I'm very, very grateful to both Laura and Nigel for summarizing so nicely so many of Riley's wonderful attributes. And I think that final one in particular, you know, I have the pleasure of having some areas of synergy with Riley's research, and sometimes we do end up at the same conferences. And, you know, one sort of personal comment I would make is how well Riley's respected at those conferences by all her peers in the community. And that respect is very well deserved, and I know is just going to keep growing over time with all the wonderful progress that your group's making. So what I'd like to do now is just call you up, Riley, for another round of applause. Are you giving flowers, Anthony? No? Or is that something else? I don't know. I just see flowers. I don't know if they're part of the show. Huh?
They were before.
Oh, these are from the husband. These are from the husband. Good. I was improvising on the spot when I saw flowers, but let's give Riley another round. Well done. You can give the flowers. Yeah, I finished. Yeah, good.
Why don't we be upstanding for Riley and then give her a final round of applause, and we are all invited to Ognisko for refreshments, further discussion and celebration.
Woo!